My name's Chris Trump. I grew up on the Big Island of Hawaii, and we grew up farming. Or I grew up farming, and my brothers. Um, we still farm. Our family farm is 750 acres of organic macadamia nuts. I managed that until about 6 years ago, um, where my brother was coming back from college and wanted to be involved, and I was like, you know what, I've done my 10 years, I'm going to let him do it, and so he's doing a great job. He's a better manager than me. I'm a better farmer than him, so I'm going to get back involved because there's work to be done. But in that job of managing, we came to need a better way to farm. We were split operation, conventional and organic. We had about 400 conventional and 350 organic. And I always joke, but it's not really a joke, that my dad created the business to provide a living wage in an economically suppressed community. So our business model is unofficial profit sharing, and is always to our staff is going to be where a lot of the costs are, and it's designed that way to there's kind of tourism, some other construction. There's lots of building, but there's very little jobs in ag, and most of our staff lives within 5 minutes of the farm. So we couldn't change how we did wages and benefits and staff, and we needed to change something about our farming practices and our efficiencies. And we were losing money in organic. Our 350 acres were operating in the red and being covered by our conventional acreage, and we had stellar organic practices. As far as that goes, we were composting, we were doing compost teas, buying fish, worm castings and making teas, and in all that, our organic was still quite yellowed and unproductive compared to where we could put inexpensive, 30 fertilizer and spray Roundup. So I began a journey to kind of ask the question, what happens in nature to create fertility where, if left alone, it's fertile? And that was a difficult question to answer with Google. But because we haven't really studied that, or as a international academic community, there hasn't been a lot of look at that. We have our kind of conventional farming practices. And I got the opportunity to hear a lecture about natural farming by a wonderful man named Dr. Hoon Park. He's, I think he's 80 now. He's Korean American. Grew up as a medical doctor, family practice doctor, or not grew up, but lived his life as a medical doctor, and had 40 years doing family practice in Hilo, one community. And he had a revelation as coming to the end of his career, where he looked around and he said, medicine has gotten better and better over the last 40 years, but people are sicker and sicker. This is his experience as a medical doctor in a farming practice. And so he's like, I need to figure this out. And he came to food. So he took a journey to South Korea because he had contact there, and he had heard of things happening with food that were really good. And he stood in a building, standing room only, kind of a warehouse type building, full of farmers. And they stood for 2 days, 11 hours a day, to hear this guy Cho Han Hugh, or Master Cho, speak. And he liked what he heard. He liked what he saw happening in Korea. So he came back to Hawaii and started just telling people about it. And so he put on, with his free time, these lectures where he taught about natural farming for free. And I went to one of these lectures, and Dr. Park is not a farmer. He's he's a medical doctor. So he's talking about cc's of this material going into so many liters of this material, and he talked about some of the other practices, and I just remember him getting heckled at one point because he's explaining this transplant protocol in natural farming, and all these tree farmers are like, "You can't do it like that. What do you know?" And he had no answer for them because he's not a farmer. He's just sharing what he learned. And we sat silently in the back. Me and my dad were there, or farming a big old orchard. And I left kind of like, I didn't really get much of that, but there's something there because I've been digging and digging for how to tend to the fertility of our orchard organically. And Master Cho, through some work of some community members in Hawaii, was brought out with his daughter for a 5-day conference, an intensive training in how to do this. So I attended that, and it ended up being the only conference in English, intensive training he ever taught in the U.S. or in English in general. There's been some in South Korea that I've gone and studied with him, but I approached this information as a farmer that needs to pay my employees and have a profitable farm very skeptically. That was my service to the knowledge, was we'll see. And so I did a 14-tree trial, and in that 14-tree trial, I had a couple trees that are what we call macadamia quick decline or macadamia slow decline, which is a root-borne fungal disease, Phytophthora, which plagues a lot of fruit trees and nut trees. And those trees, once they defoliate, start defoliating, we know they will never come back into production. Within a year, if it's quick decline, or a few years if it's slow decline, will be things we cut down and push into the push pile. But I applied indigenous microorganism, which is one of the preps in natural farming, which I'll explain here right after this monologue. And a few other the preps, and I watched it for a year. We had a lot of farming to do, so I wasn't tinkering around with it. And in a year time, my Phytophthora trees refoliated. And for me, that was a big deal, because there is no answer in our industry for that disease. The only thing that they had was some experimental work out of Australia, where they spray the whole trunk with phosphoric acid to get it to carry into the cambium layer and down to the roots to kill these bad fungus, and that wasn't very effective, and the tree suffered. But I had just seen refoliation in one of my disease trees. And then I was like, okay, we're going to go for a 5-acre trial. I did a 14-tree trial, and I found out that they were making it. You don't understand what it is they're making yet because I haven't explained it, but bear with me. And they're making this inoculum, this soil inoculum. And I was like, great, I don't have to make it. This seems kind of challenging. I had learned at a wheelbarrow scale. And as farming acres, I was like, okay, a wheelbarrow at a time would take me about 30 years to go one round. And so I was like, great, they're making it. So I ordered 20 tons. And I applied it on a bunch of it on 5 acres. And within a week, all my trees turned bright yellow. And I was like, oh, I just killed 5 acres of highly productive trees. This is not good. They're 30-year-old trees, this is significant. And right about that time, I'm like, okay, what's going on here? What happened? I got the opportunity to study with an innovator, or kind of a original advocate in microbial farming, Dr. Elaine Ingham, And she taught me the use of the microscope and something really key, the concept of successional fungal bacterial ratios, which is the diversity or the balance of different species of microorganisms in the soil. And so day one of the class, I bought a microscope because we're a multi-million-dollar farm, and I was like, we need to answer this question. And so I started, first thing I did was throw some of that 20 tons of material I bought under the microscope. And what I discovered is that it was all bacteria. There was no fungi in there. And because of the way they were making it, they were doing it with a boiler, heating it, and causing activity in 24 hours and then calling it finished. Well, fungi doesn't grow in 24 hours. Fungi is a week-long process to really multiply. So they were excluding fungi, and my trees, though they needed more microbial life in the soil, had struck some degree of balance before I put that on. Trees like a 30 to 1 to 1,000 to 1 fungal to bacterial ratio, so heavy on the fungi, and I had just put down this massive load of inoculum of solely bacteria. So my trees' reaction was to me disrupting think of their gut flora balance, and nothing else. That was it. There wasn't a toxin in the material. It was simply that I had disrupted the balance that they used for eating, and that didn't stop me though. I was like, okay. I realized that if I'm going to try this, if I'm going to do it, I need to actually understand it. I need to understand it in a way that my teacher's not able to teach it to me, because this is traditional knowledge. He's incredibly intelligent man, this Master Cho. Master is the word like sensei in Japan. It's teacher. It's a term of respect. It's not like you are my master. It's more like teacher. And so Master Cho studied enzymatic theory in Japan, and with a bunch of really prestigious Japanese doctors at the time. He speaks Japanese. So super knowledgeable, but not able to explain all the whys. And I needed to know so that I could know how to scale it, because there were no teachers on how to scale it. Our farm was the first to scale Korean natural farming on a large scale in the world. I needed to understand what parameters were shiftable, in order to know that, I needed to understand how it worked. And so I began a long journey of side-by-side trials and blood, sweat, and tears, and we made enough for 5 acres of inoculum, treated 5 acres now with a fungally rich IMO, and we're watching it. And we had the following year an 8-month drought event, which in Hawaii is a really long time to go without water. We don't typically see that, not where we live. And so all our trees yellowed. They were all experiencing water stress, except for my 5-acre trial and my 14-tree trial. We were driving through the orchard, looking at the trees, kind of assessing, and we came across 2 green patches in a water drought season. And for us, that was enough. For me, that was enough to say, okay, that's reason enough to continue. If I'm buffering against drought stress, I can take the next step. And I did a 50-acre trial, and then I ran a 144-acre trial for 4 years. And by the end of that trial, we looked around, assessed our costs, and said, yeah, this works. And we converted our farm over to 100% organic and used natural farming to tend to the kind of nutrition of our trees. We still run fully organic. I left 6 years ago, trained staff to do it, and for 2 years he was great, and then he stopped working there, and my younger brother's like, I don't know how to do this stuff. I'm not continuing with it, so now I got to go back and help them restart. But it works. Since then, I've been all over the world working with farmers in different sectors in ag, golf courses that run no more fungicides at all, no more pesticides, and don't deal with fungal disease. Golf courses have fungal disease endlessly, and the natural farming golf courses don't deal with fungal disease at all. And work with grape farmers. I was just in Portugal a week and a half ago, working with wine grape farmers. We're doing the University of UTAD University, which is a local Portuguese university, and helping deal with downing mildew in wine grape production, which there's not really an organic answer for. And work with row crop farmers in Iowa. One of my clients is 1,000-acre corn, soy, alfalfa farmer, and he's doing it all with microbes, and just stopped, completely ended. He's 6th generation on that land, and he's doing a really great microbial inoculum in-house fertilizer production program for those crops. Though corn is tricky. Corn, you got to spoon feed in transition because the soil's not cycling enough nutrients for it. But learning things. For many of you who have no idea what I'm talking about, let me explain. Fun to have a tiny bit of background. We did that 144-acre trial, and I had macadamia nuts. A pretty young industry. It's like 80 to 90 years old. Really in commercial production, maybe 80 years old. And so we have like this. Some of these guys that are 65, 70, getting into retirement, and they have been experts in the macadamia nut industry for that whole time. They're the OGs of the industry. And so one of those guys came out, and I explained what I was doing. I grew up around him. He's a friend of my dad's, but just really knowledgeable in macadamia nuts. And he looked at it and he said, "There's no way. You can't." Because I was talking about farming, maintaining the same production while using 1/100th of the recommended fertilizer for my crop. And he's like, "There's no way. You'll not, this, all the reasons it's not going to work." And so we kept the trial going. And so 4 years later, he brought the head of the International Macadamia Nut Society out to our farm. It's kind of a big deal in our industry. And they walked the orchard, and at the end of it, the head of the International Macadamia Nut Society said, "We would like you to be on our research and development board and speak to the industry once a year, and because these are the most beautiful trees I've seen anywhere in the world." And so efficacy is there for me, and I teach this because somewhere along the line, initially I was just trying to make our farm more viable, but somewhere along the way I came to believe that this can be an answer for a real problem we have in food worldwide. And I still believe that to be true, and so we're plotting world takeover now, and succeeding so far. So thanks for being here. You can join the cult after with another QR code. I'm just kidding. It was a joke. All right. Now you get to see my epic drawing skills. This is not why they invited me here. So this is a tree. Don't go by what your eyes tell you. Listen to me. I'm telling you it's a tree. Or a plant. We'll call it a plant. And as you know, or maybe you don't, any tree or plant you see typically has a equal footprint below ground as it has above ground. So, one of these tall trees, if you flipped it upside down and we saw just the roots coming up out of the ground, it would look somewhat similar and have a similar kind of canopy, if you will. So one of the philosophies in natural farming is that the plant above ground represents the plant below ground. So this is an outcome of this. And we're coming to understand that as an academic community too, that this is the case. We all learned about photosynthesis in biology in high school, right? Sunlight shines on the plant. The plant's like a solar system. It takes in sunlight, uses water, and creates carbohydrate, which it uses for its structure, its leaves, its fruit, etc. Right? Well, all plants take some of their carbohydrate that they're not using for their fruits and their leaves, and they push it down into their root zone and create an exudate field, a field of nutrients that they produce. And what they're doing is they're opening something like a bakery. They're opening a bakery full of sweets. And the currency to buy their baked goods is boron, zinc, manganese, iron. They trade in their root zone their exudates, their excess, or not excess, but planned carbohydrates. They trade it to microbes in their root zone in trade for the things that the microbes are processing in the soil. So fungi is able to eat sand, silt, and clay, or digest and break down rock. So you might have a fungi over here, and it's 100 meters away, working on granite and sand and whatnot, and it's also touching this root zone. And in the root zone, there will be an exchange of minor nutrients from the plant, carbohydrates, for minor minerals from the fungi. This process and this zone where this is happening is called the rhizosphere. I won't write it down. You guys can look it up later. The rhizosphere, and this process is rhizophagy. And what will happen is a bacteria can use a fungal hypha, the tube. Fungi are tubular microscopically, and they can use it like a subway system. Bacteria will travel through a fungi back and forth. And so over here, the fungi is chewing on and digesting rock, sand, silt, and clay, and bacteria are symbiotically working with the fungi. The bacteria get a sheath, or kind of like we'd put on a jacket, of charged nutrient material. And they'll come back to the root zone, and the roots will blast them with a little bit of oxygen, and it sheds their jacket. And so the plant gets those minor minerals that are being processed, and then the bacteria gets sent out through biochemical communication with the deficiency or the thing that the plant's going to need based on the biocommunication of the exudate field. So the plant is saying, I'm hungry for this, and the microbes are saying, well, you are our bread and butter. We will feed you whatever you need because you keep giving us all that good sugar. And so it's a symbiotic relationship that happens in the root zone, not just with fungi, but with the entire soil food web. There's things that eat fungi that then produce plant available nutrients. There's a infinite symphony, if you will, of interconnectivity happening in an inch of soil down in the root zone. Yeah, question.
Where does the boron, zinc, and manganese come from? Is that
That's an excellent question.
In the soil?
Excellent question. So a lot of people I've encountered think of this as, this is the thing that holds the plant upright, right? This material here is the thing that the plant goes in. I need to add the fertilizer and the water so that the plant grows. Well, that's not the case. This is the fertilizer, the bag of minor nutrients. This is all of that. This is hundreds and hundreds of years, thousands of years, maybe an infinite resource of all these minor minerals, but they are trapped in complex bonds that plants cannot eat. A plant root cannot break apart a rock. Other than the plant roots have minor acids they can use to split a rock to seek water, but they're not digesting the rock to eat. They can simply move through it. But this is non-plant available minerals. These minerals, some of them are water soluble, but you got to figure if all minerals in the soil were water soluble and plant available, when the rain comes, they would all wash away. This is nature is like this. I don't know. The deeper I go and the more complex I see nature, the more I believe this is painted and designed to a symphony level. Everything playing its note in unison. But for fungi, this is chewable material. Fungus is how I live in Hawaii, and everybody says, "Oh, volcanic soil is, rich and wonderful for growing plants." Well, no, not without fungi. Volcanic areas are, but not without fungi. Because without fungi, it's just rock, and we got lots of rock fields. That one time I was driving with Master Cho. We picked him up from the airport, and he's doing like a 2-day talk in Hawaii. And driving along, and he tells through Dr. Park, "Chris, you should buy this land we're driving past and farm on it." And I look out my window, solid lava rock. Scratched my head, and it wasn't until like a year later where we had been processing rock, and some people had run these trials, building topsoil on lava rock. That I was like, with high amounts of diverse fungal communities, you can actually process these inert materials much faster. So there's a whole system for that, for processing poor quality soil into fertility in this system. But back to my beautiful drawing. As this process grows, all the other things in the area start to increase. So worms breed based on food source, right? There's many scientists that believe that the count of worms there are in your area is the best metric to know how fertile your soil is. Just number of worms, number of earthworms. But worms breed based on food. So these are bacteria I'm drawing here, just in case you needed. There's some fungi. Worms breed based on food source. So the more, and what worms eat are microorganisms in the soil or in their area they are. They eat bacteria mostly. And so they'll pass soil through them and eat all those yummy proteins that are in microbes. Because microbes are like us. They're 70% water, nitrogen, etc. So mommy and daddy worm get together and they say, how many babies should we have? And they make that decision based on how much food is there. Well, if there's a thriving microbial food web, then worms go ballistic and multiply like crazy. So, and then earthworms, not red wigglers, those are composting worms. They function in the compost pile. But in the soil, we have soil worms or night crawlers or some of the various species, and they typically live 18 inches below the surface. But they eat, because most of the microbial life is near the surface, they eat up top. So worms go up to eat and down to live. This process, as the worms increase, you're increasing water retention, soil arability or oxygen transfer in your soil. So as this grows, the whole symbiotic system ramps up. And this is where these plants... Have you guys had a strawberry from like Costco or one of the big stores that was big and beautiful and red and totally tasteless?
Yes.
Okay. And then you've had also a strawberry from Grandma's like Strawberry Patch, and you bite into it and it's just like an explosion of flavor. Okay, so what's happened there? Well, a while ago we learned that and knew that was absolutely true that all plants need to survive are three things: nitrogen, phosphorus, and potassium. That's all plants need. And so they sold it to us. And then we had the Dust Bowl, and they're like, wait, actually there needs to be 23 minor minerals, and on and on and on the products we can buy, right? Well, that strawberry that's tasteless is based on that principle. Plants just need NPK, so they can grow them hydroponically. They put pink and green nutrients in, and the plants grow big and green and red and beautiful and totally tasteless. Because for a plant to produce those complex sugars that we associate with flavor, they need complex nutrients. They need materials to be able to put together to create those complex sugars. And in an NPK limited fertilizer, they don't have all that diversity of minor nutrients. And so the nutrient density of that food is quite limited. Whereas in this scenario, this plant is actually getting on-demand line in of their minor minerals. They call for and get from the surrounding area. So I'll give you an example of this. I started farming like this. We're running our 144-acre trial. And like a good farmer, we do our soil and tissue analysis every year in the same areas. So we have kind of a snapshot of how things are changing. And I remember as a kid, we'd get back the recommendation from the extension agent. And every year it was, 1 ton or half a ton of calcium per acre to fix our pH problem, and after 10 years I'm talking to my dad. I'm like, Dad, I don't think this is working. Because we always have the same recipe to fix the pH. It can't be solving the problem. But as we're running, that was totally unrelated, sorry, that was just a rabbit trail, the calcium. As we're running our trial, we're still doing our tissue and soil analysis, and so we ship off our samples. Most of our samples are typical, where we're conventionals, orchards are still conventional, our organic still run organic. But then the 144-acre natural farming trial, we sent away and we got back. He wanted to have a conversation. He's like, what are you guys doing here? And we're like, why? What do you mean? He says, well, you have the same soil deficiency that we're seeing throughout the farm, or that you see historically in this block, but I'm not seeing the deficiency transfer to the leaves. What's different? What have you changed? So we're explaining to him we're, inoculating with this highly fungal inoculum, diverse microbials, yada yada yada, and so to re-explain what what he's seen is he's seen this deficiency, this soil test deficiency. We have too little manganese, or insert any of these soil components, and so that should be too little manganese in the leaf tissue. It should be it's generally a direct transfer. So that's how this works. Most of agriculture functions like this. Farmers don't know how to tend to their soil, other than they send the soil tests in, they get a readout, and they get a recommendation back of the fertilizer to buy to balance the deficiencies. This is how most of America farms. What happened to our sample in this now microbially rich environment is that we had the soil sample showed that same deficiency, but somehow the tree had everything it needed. And so next year, when we sent the same soil analysis away and got back our results, he had on our 144-acre trial sections, he had an asterisk. He's like, you may need this and this much, amendments to correct this deficiency, asterisk, unless high amounts of soil diversity or microbial diversity are present. Now all of a sudden this whole protocol of getting the minerals we needed to, or the nutrients we needed to correct our deficiencies, didn't work. It no longer functioned in this, because my theory, is that now our plants are getting access to that manganese that was tied up, not available on our soil test, but they're getting it trickled to them through this symbiotic relationship with fungi in their root zone. Does that make sense?
Mm-hmm.
Tracking with me? Why am I talking about this? What does this have to do with natural farming? 80% of natural farming, or what I think of as 80% of the benefit of natural farming, comes from this process we call IMO. I apologize ahead of time. I will spit out some acronyms. Please grab me or raise your hand if I have not explained it. But it's a mouthful to say some of these words over and over, so we use acronyms. It works, but initially it's like you join the military and you don't have a key. So this is indigenous microorganism. We can put an S on that, but IMO. Ooh. Yeah.
Defining, when you use indigenous, does that mean like endemic, like to the area, or ancient, or?
Indigenous to the area. Indigenous to your bioregion, your microclimate, your barometric pressure, rainfall, general climate, soil types. So yeah, within, generally, more or less a 30-minute drive, around here is your general ecosystem. That doesn't mean you can't go a little further, but that's generally what I'm talking about. So you guys have seen the huge increase in the last 10 years of biostimulants or bugs in a jug, microorganisms in a plastic container, to very expensive. They work. Very effective for cycling nutrients in your soil. Lab-grown microbes, and I'm an advocate. I think they're a great tool for a farmer. And they are not indigenous. They're grown. They're selected for their qualities, something that they isolated, that they do in nature that is beneficial. A lot of tout nitrogen cycling or nitrifying this or that. There's a bunch of things you can buy that tout this or that effect. The problem is that the testing shows they have about a 6-month fall off, which is great if you're selling bugs in a jug because you need to buy it a couple times a year. Again, nothing wrong with it, but it gets outcompeted by your indigenous microbes in about 6 months, and it just doesn't exist there anymore. And so what this process does is it teaches you to be able to go out in your wild places. Ideally, we're looking for untouched forest or places that haven't been disturbed by humans for as long as you can find. 1,000 years would be great. But just places that haven't been disrupted as long as possible. And we cultivated on rice. So it's like an auger, the petri dish in biology class, you grow some bacteria on. This is like that, but for fungi, because fungi don't eat those simple carbohydrates like bacteria do, that you find in an auger. They actually, their food is fats. They take fatty acid chains and they use the energy in tearing them apart. They consume the lipids and they reconstruct the acids into fulvic and humic acids in these complex, super long acid chains. But rice has one of the best fat protein profiles of any grain in nature. And so we put it out. Fungi and yeast and bacteria, a whole kind of community of microorganisms bloom on it, and then we take that, We mix it with an equal weights brown sugar to put it into a dormant state. So we're taking a snapshot of the forest's microbial life, causing it to go dormant, all the bacteria that can cyst and the fungi sporulate. And so you have seeds of this culture. Think of it, somebody was talking about it like a sourdough starter earlier today. Sure, those correlations work if they help us understand it. It is a simplification. Sourdough starter is a symbiotic culture of yeast and bacteria. This has the fungal community in it and potentially protozoa that can cyst and go dormant. When you go on, I'm going to preempt a question. Why do you use sugar, Chris? When I heard they landed in Cape Cod before they went over to Plymouth. Is that true? Yeah? Okay. So on their long sea voyage, they wanted to bring some fish. And if you bring fish on an unrefrigerated sea vessel, you're going to have some stinky fish after a few weeks. So what would they do?
Try it.
Salt.
They'd stick it in a barrel of salt. Why? Why did that stop all rot? Because salt is dry, just like a paper towel is dry. And that dryness chemically looks like a nice shape for a water molecule. So what happens when water came into contact with that salt barrel of fish is that all the water that came in got captured in chemical bonds with salt molecules and wasn't available for use by microorganisms. So it is the osmotic pressure or the dryness of the salt chemically that actually creates a scenario where it's desertified or dried out, even though they're surrounded by water. Inside that is so dry that, and microbes are like that. You want one of your workers to work for 2 days without water, good luck. The same thing. Microorganisms can't do any of their processes without water molecules. And so that's what we're doing with these microorganisms. We're taking them, they've bloomed, we got them all lush and eating and thriving in this culture, and then we mix them immediately with brown sugar because we do not want any generational changes from that initial snapshot from the forest. So we don't want them to keep going like this and multiplying and dying and multiplying and dying, because you'll get a skew or a change because now they're out of this environment. So we put them in cryo freeze, to use a sci-fi term, to cause them to go dormant. So now what I have as a farmer, and this is what I think is the best single piece of natural farming, if you take away one thing, is I have an inoculum on my shelf that's shelf stable for 3+ years. And so what I do then as a natural farmer is I grab snapshots of these wild environments in spring, summer, fall throughout the year. So I have like 9 collections from 3 different places throughout the year. And now I can grow those out on a substrate. We use oats and rice bran. Yes, question?
Yes, so the sugar is acting as a desiccator in the same way as salt.
Exactly right.
Can you just explain why not salt?
The sugar, I think, is slightly gentler on the living organisms, and the food does have or the carbohydrate does have a minor role to play in the process. Yeah.
How about if you use like a typical brine mix of sugar and salt?
I think that could definitely work. What I teach my students, because this is a complex process that has hundreds of years of kind of data, is to practice it just the way the recipes are. Learn that, and then if you want to, because this is pretty. I'm gonna. Can I give you a hard time? Can I use you as a? Okay. It's a very American thing that we do when we learn something, and I think it's one of our strengths. We're innovators. We forge new ways, and so when somebody's teaching, before they've finished explaining a process, we'll come up with a new way to do it or away from something we already understand. And everything I'm explaining to you is pretty much new. I just encourage, kind of take it in as it is. I love innovating. I am definitely an inventor, and there's things that we have discovered and ways we can use this and variations. However, these processes are quite good. Over and over I dig into these natural farming practices looking at the scientific basis, and over I'd find solid science. Because, and I was explaining this at the with the nonprofit earlier, what is the definition of science? I'll give somebody extra credit if they got this. Ooh. Investigatory is a good word. I'm looking for 2 steps. Empirical method.
Observation.
All right, that's one word. Observation and testing. Yeah, experimentation. So science is the study of nature through observation and experimentation. Who are the best scientists before we had the current academic system? Farmers are in nature. They're trying something, it fails, they nix it. They don't teach the next generation that failed thing. So every generation, they're passing on anything they learned that was effective for producing crops. And what is it that they're coming into? They're coming into alignment with natural law. The things that work with natural law, before we're hacking natural law with chemicals, the things that worked with natural law are the things that were carried forward in farming systems. So these thousands of year practices are carried on or transferred to the next generation through love and care, because you don't teach your kids the things that failed, you teach the kids the things that work. And so as we look at some of these farming practices, even though they're old, antiquated, before we had microscopes, etc., it wasn't necessary for them to understand all the why to know that it worked, because that's what they were doing, right? They're experimenting. This is effective over and over. We're going to keep doing this. So a lot of natural farming isn't anything new. It's just long-held ways to farm that now we're discovering. Actually, that is cutting edge. This is the cutting edge of what we're seeking in science, how microorganisms. I met I'm rabbit trailing a bit. I'm sorry, guys. I met a guy who ran a lab in San Diego, multi-million-dollar lab. And we were chatting over shave ice in Hawaii. We were just randomly having a conversation. And he is explaining to me that his whole lab and his whole staff have been leased for several years now by Monsanto. And they are, their whole job is to seek how microbes make fertilizer. Yeah, you want to be scared. You want to be scared of something. Don't be scared of GMO. Plants are going to express whatever their soil's in. Be scared of GMMO, genetically modified microorganisms. That will be what turns the world to zombies. Zombie apocalypse. No. All right, where are we? So IMO has 4 stages. I just explained a couple of them. IMO 1 is that nice rice box with a bloom of fungi on top, right? And then IMO 2, we put it into a container, half-half, with brown sugar and microorganisms, okay? And the reason we do equal weights is because that accounts for all the weight of the water in the microorganisms' bodies. So the dryness of the sugar will actually desiccate the microorganisms, where they'll start drying out. And so their response to going into desertified environment is to go into their dormant states, which we all know that fungi have spores, and that the spores can survive for how long in icebergs? We don't know. So my point is, if you've effectively caused them to go dormant and you care for that, it doesn't get disrupted or in sunlight or add water to it, that actually is a shelf-stable inoculum where that seed of that organism is actually ready to be sown in your field. Yeah.
When you're mixing the rice and brown sugar and letting it go dormant, is it like a 50-50 mix? Is it like
It's exactly what it is. I'm giving an overview of some concepts. I have 25 free YouTube videos that go through each one of these processes. If you want to know more, there's a website, there's online community. This is internationally growing quite quickly, and there's a lot of information becoming available in the English-speaking world. So you definitely can get deep into these practices if you so choose. I am teaching in Truro, a 5-day intensive class to train people how to do this. And one 5-day class, you don't need anything else. You have everything you need to know to do it. So it is really accessible, but there is a learning curve, which is what we try and help in the 5-day class. Yeah.
2 quick ones. What do we Google to get to this? And the second thing, are you teaching at Castle Hill? Can we find out about your course through Castle Hill?
I don't know who Castle Hill is. No.
Oh, it's an organization out that way.
Nope. It's my own company. I am here because one of my students invited me out, and so I'm teaching kind of his community
So
of
what do we
people.
Google?
I'll write all my contacts in just a minute. So you can definitely Google. If you want to Google on YouTube, you can Google Chris Trump, just my name, CHRISTRUMP. No relation.
Thank you.
So IMO 2. Wait, did I not answer the other question? What was the other question? No, we got it. Okay. IMO 2 is in cryo-freeze. IMO 3, we take this inoculum and we grow it out on a substrate. My favorite materials are rice bran and oats, kind of a 50-50, and then that mix with 50% wood chips. And you put diverse nutrients in there, some vinegar that buffers pH, and you manage it under 120 degrees because all the microorganisms will thrive between the 100 to 120 degrees. You don't let it go hot because you get very skewed or narrow in your diversity. And at the end of 7-ish days of turning a pile, IMO 3 is carbohydrate, which is the oats and rice bran, and this is carbon, so wood chips or straw, and then you turn. That's a 7-day process. This usually takes 5 to 7 days, but you're turning it kind of regularly. The next stage is IMO 4. So when IMO 3 finishes, what we're doing is we have a pile, right? And all our substrates in there. When I come after a few days of growth, I have this layer of highly fungal mat in the top 4 inches of the material, just solid fuzz and striations. And then it's kind of heating up. I need to turn it to keep it under temperature. And so what I do is I'm taking that material and I'm churning it in. So some of this mat that's on top ends up in a chunk in the middle, right? What happens to that? Well, it's a little too hot for a lot of your big aerobic organisms. They love that outside, slightly cooler, the fungal like that slightly cooler, highly aerobic environment. And so they bloom on the surface, and when they go to the center, they sporulate. And so we're turning it, we're getting this bloom on the surface, and then we turn it inside. Now some of those fungi that were blooming and multiplying or cruising send their nucleus into a fruiting head and sporulate. By the end, they've used up all the water I initially added. This is dried out to pretty dry material, and they stop working because they've run out of water. Now, if I look at it under a microscope, that whole pile looks like a bag of marbles. It's all fungal spores. What I've just done is I've taken that small inoculum. I might use a handful or so of this to grow 200 pounds of material out. And now I've taken my microbes, or my area's microbes, the indigenous microbes, and I've grown them out on the substrate. The next stage, IMO 4, is I mix my IMO 3 with equal mass soil from my farm or from the mountain nearby, and ideally this is somewhat alive, not like structural fill. This is soil, not dirt, and that process begins again. I mix them and keep it under temperature. And what I have in my soil, here I have bacteria, yeast, fungi mostly. Here I have microarthropods, nematodes, amoeba. I have the whole kind of protozoan mix of my soil environment. A lot of those guys eat fungi and bacteria or use fungi and bacteria. So I create a party, I introduce everybody, they all find balance, and then that finished material I can apply straight onto a growing sensitive crop without any shock. Because I've already used the soil to let them find balance where there's tons of food and moisture in this IMO 4 process. Yeah, question?
2 questions. One, does your wood chip or straw content make a difference? Like the kind of wood, the acidity, whatever?
So, yeah, you can never use American redwood. You just can't. It's too antimicrobial to stop everything. But other than that, you can use most wood chips. Straw, you just need to have it needs to be like 2 to 3 inches pieces. You can't have big long straw. It won't be aerobic enough.
And then the other thing is, when you're talking about in the IMO 4 merging your inoculant with your soil, is that a combination of like dirt and compost? Is that
So this is very specific. IMO it is a
I mean
type.
the soil component?
Oh no, the soil, you want soil. You don't want soil and compost. You want like dirt. You want this here.
Okay.
Yeah, and you want, ideally you get, I go more into this in other places if you want to dig into it, but you want some from maybe a higher mountain or an area. Ideally you want 3 components. You want your farm soil, some from a little bit higher, and you want some red soil if you have any. I don't think you guys have red soil out here. Yeah. Red soil is high in iron. You don't need it to do this process, but it does create some value. This is way too fast to explain any of this, but I want to tell you a bunch of things, and we're just cruising through in 3 hours. So we're going to show you some things. We're going to go. I'd like to cruise right now after a few more questions and wrapping up this explanation. I want to show you what I'm looking for when I go out into the forest to find IMO 1. I want to show you the mycelium and why we're selecting this and not that. Yeah.
If you're familiar with the book, The Regenerative Grower's Guide to Garden Amendments, and if so, if this is a similar process. I know there was one that involved purchasing bran and having a tarp.
Is that a new book?
Is it new?
Yeah.
I'm not sure how new it is.
Because I know, is it Nigel Palmer?
I think so.
Yeah, so he basically watched my YouTubes and added it to his book. Yeah, I was just given that in Ireland last year, and he just learned it from us. Then we learned it from Master Cho.
Different things, but this sounds familiar.
Yeah, no, he added a bunch of really cool organic practices, and then he just added natural farming to his book. Yeah, it's the same, but he has a couple things wrong.
Do you want to elaborate on the wrong things?
No. We'll nix that from the video. Edit that out. I'm cheering for his book. I think it's really great that he did it. It seemed awesome. I haven't read it, but we reviewed it with a friend that had one of my students, and we went like, ah.
Sorry I brought it up.
No, it's great. It's a good book. I recommend buying it. Yeah, no, I'm not knocking it. More and more you will see this in literature. It just hasn't been. This came to the English-speaking world in 2011 when we started sharing it. So, yeah, it's new, and people are going to incorporate it. And there's nothing wrong with incorporating it into your farming practices, like using some of it. There's a whole system, but everywhere I've gone in the world where natural farmers are using natural farming, they use what they use. The best of them, they all use IMO, but then some of them use other aspects and some of them don't, and some of them, change some things. India, their natural farming looks a little different. Philippines, they've adapted things for being ultra cheap. They just won't spend any money on any of the preparations, and it's what it is. Ultimately, I think the best part, my favorite part of natural farming, if somebody really gets it and starts to see it, the observation kicks in such a way that it's kind of like a key for understanding nature. You're like, oh, nature's doing it like this, and you do all these things, and you're like, wait a second, I'm just literally emulating, copying what nature does, partnering with natural law to grow food. It's not some, this isn't some advanced tech. It is the representation of years and years and years of observation and experiment, the study of nature, observation and experimentation. And so, and then we're studying it. We're now doing research studies and seeking to understand it a little more. And so tiny tweaks come in that are just, and Master Cho told me, we were riding in the bus in Korea, and me, him, and Dr. Barker were sitting together, and he said, natural farming is not finished, Chris. You got to finish it. And he wasn't talking just me, but the next generation. He's saying this is, and he brought together other tech. He brought together this, long tradition of Japanese studying microbial farming, along with these long-held traditions of Korean farming, and then he developed some things. He also would credit God giving him dreams, and he'd wake up and he'd try it, and it worked. And so this came, and the reason I like natural farming as a commercial grower is because it is an elegant method. There's a lot of traditional things that are done around the world that work. But this is a package that, with a week of training, my friend Michael went through training and then came back and became a trainer. And there's people all over. But with the week of training, you have everything you need to do it. And there's no products to buy. There's no subscription to subscribe to. It's kind of a sovereignty for farmer movement. And some people, I work a lot remotely in Africa, just helping through dialogue. And some of these farmers, they started with nothing. This one guy is so proud of his accomplishment, and he should be. But he's always kind of, look at that. Is that osprey?
Yeah.
Massive bird. So pretty. He started a 3,000 pig piggery and an 8,000 chicken farm. And he's a wealthy man now in Africa, and he's just done it through this process. So there's the whole livestock component of natural farming, no smell piggery. I know it sounds ridiculous, but it's real. Yeah.
I was wondering how you scaled this. You said you did 144 acres. So.
We do 750 acres. Yeah. One of the ways, just a process. I teach. It's nothing tricky. It's just. But one of the advancements was liquid IMO, being able to brew it out like an actively aerated compost tea, and spray it out. The cost is really low. So I would spray 3 times a year. and 3 times a year spraying liquid IMO cost me $27 per acre per year. And that was for the labor to make all the stuff, any of the material I bought, the labor to apply it, and the tractor hours to apply it. So yeah, it's pretty inexpensive. And that's really, people ask me, well, what's the benefit? I'm like, well, there's a lot of benefits. The potential, most natural farmers don't deal with disease anymore. It's just not something that's part of it, because you have so much diversity, and diseases are typically opportunistic, so they function in a vacuum. Where there's nothing, they go ballistic. So most natural farmers aren't dealing with disease. Bugs, if a plant is getting complete nutrients, they are able to formulate total proteins or complete proteins, and they put those into their cell walls. That becomes like one of those hard to open plastic packages that your little like USB card comes in. You're like gonna kill yourself trying to cut it open. That's what a complete protein in a cell wall is to a bug that wants a little juice. A plant that's been pumped full of chemical fertilizer has so much juice in it, just ready and bursting. So the bug just pokes it and it gets a bunch of sugars. But if there's complete proteins in the cell wall, typically bugs will look for an easier meal. Yeah, question.
I was going to ask, because you said the microbes in a jar, you said they last maybe 6 months. When you create some kind of like homeostasis through this practice, since it's indigenous, does it
Self-perpetuates in the soil. Yeah, that's kind of one of the cool things is I would say in doing this for 3 years, once you get everything kind of alive, you never have to apply microbes again.
So it could be like a couple times.
Yep, yeah, 3. I mean, I say for soil to really change in a commercial farming system where you're still working it and doing all that stuff, I say 3 years, but really you'll see effects immediately. It's just for it to really, there's a sweet spot, a total change where you're like, I don't deal with bugs anymore. This is a typical thing in 3 years, but immediately you have, I was gonna say, people ask me what's the benefit, there's disease resistance, etc. But our farm, we stopped being able to herbicide, which was a huge efficiency for us. There really was cost savings, and which is great. We don't like herbicide. We were happy to get away from it. I hated having an employee, Anyways, but now we have to mow more. So we gained cost. We lost money, if you will, in losing herbicide. But we also saved money in we spend a whole lot less on fertilizer, inputs, etc. Well, so I say that we maintained our yield. Our yield stayed about equal. We gained a cost in labor and mowing, but we added a job, which was within our ethos. And then we saved a bunch of money in inputs. And so I say we farm for about the same cost that we farmed before, but we're organically certified and we get a better price. But I would say that somebody that doesn't have that huge herbicide change, that you can make more money. Like David DeWitt, who's the host of the farm I met in Truro, one of my students. He was 30 years organic vegetable grower, made his living growing vegetables organically there, and he says that he now puts away the money he saves using natural, because he had his numbers down. He knows his numbers for many years. He says he saves, his first year, he's like, I saved $2,500. I'm using that for vacation for our family. So now every year, his natural farming savings is his vacation money for his crew. I'm biased. I think we have the best tasting nuts in the world.
Do you continue to inoculate several times a year?
Right now we don't. Yeah, right now I don't. I think we could in the fields that I applied solid IMO. I don't think I ever need to go in there again with inoculum. But in the fields that I shifted to liquid IMO only because the cost was drastically less. And I would say that would benefit from me continuing to go in there. I feel like it didn't establish in the soil as effectively as the top dressing did. And IMO 4 is applied at 330 pounds per quarter acre. But this is the most of what I teach in 5 days is kind of the taste, touch, feel of going over. So this is the biggest learning curve in natural farming, is making this right. It's like making a souffle. It's got to rise, and that level of little bit of detail is there, where you kind of it's like having a baby for a week. Yeah, question. Yeah.
Yeah, so the solid IMO, is that the IMO 4?
So it's 330 pounds per quarter acre. It's like a dusting, like you'd spread cover crop seed. Yeah, it's you're just inoculating your soil.
And so the liquid is it like a tea? You make a tea
Mhm.
with that IMO 4?
Uh-huh.
Okay.
Yep. Exactly right.
When you said, I forget what it was, but the thought is, as you're doing this, is there some clear signs to see that it went wrong? Like
You can know that it went wrong in the process, for sure. There's definitely a right and a wrong way to make this, but if you make this properly, then yeah, it works. Is that what you're asking?
Yeah, like in other words, is there a part where it would just go bad and you're like, okay, I messed up somehow, I just have to trash the whole thing and start over?
Absolutely.
Or, and
Absolutely.
Is it kind of clear
Absolutely. Yeah. Yeah, it smells like ammonia when it gets too hot. Or it's the temperature. If it goes over 123 and it stays like 125 to 130 for over an hour, you'll start producing actinobacteria. They're an important part of soil, they're a vital part of nature, but they're like a monoculture, and they will turn your entire thing into a monoculture, and you'll smell all your nitrogen gassing off. They love that high heat. It'll be really hard to get the heat down. And so typically if you get over temperature and you start smelling like ammonia, we're like, you kind of failed to produce diverse microbial life. So that's kind of a failure. Yeah.
You said you took like 3 samples in spring and summer and a fall sample for the initial IMO 1. Do you then use the corresponding seasonal sample at the application time? Doesn't matter?
No, what we do is we take all those samples. So say I have 12 samples from 3 years, or however many that works out to. I'm taking all 12 handfuls from each and putting them into this next step. And because that diversity, they will establish balance within there. The key is diversity. And even if you don't do natural farming, if you're just an organic grower, you're like, I have a good practice, I compost, etc. Bring in microbial diversity. Get a handful from the forest, and everywhere you go, put it into your compost. Soil diversity, microbial diversity in the soil is the key to real soil cycling, nutrient cycling in your soil. All right, guys, we're gonna pause. Yeah, one more question. Last question.
Kind of unrelated. So if you stop spraying Roundup and you added all this like nutrient diversity, did you have like weed problems, and do you deal with them manually since you're not spraying? Like, I'm just kind of curious.
Yeah, good question. Yeah, the one thing that I don't teach in natural farming is how to kill weeds. Yeah, so, however, like I said, on our farm, our weeds now we had to mow, and a 10-foot herbicide strip on our farm saved, so now you have 400 acres that nobody needs to mow for 4 months, 5 months, 6 months. Now we have 400 acres that takes 3-4 guys to mow on circuit. They never stop mowing on that same land in that same time. So yeah, that weeding, our weeding, that's the real cost. But there's enough offset to that cost that you're like, I traded Roundup for weeding. I added an employee, and I'm still making more money.
Trying to be organic anyway, then right.
You need another employee now, but you now are spending nothing on inputs and you're getting an organic price. Yeah, so you have to play with it. And they were asking how you weed here, and some of the organic growers, they just use a flame weeder. You just pass it by long enough that it curls the leaf and that it dies above ground. You're not killing the roots, but it costs you very little. It's low labor. There's other ways to deal with weeds, and if we hadn't advanced over the last 20, 50 years in chemical farming, we would have advanced in robotic weed pulling. So now we're catching up because we're trying to go organic. All right, guys, we're going to pause on this. What I'd like to do is get yourself a drink or whatever, maybe a 5-minute break, 10-minute break, and then we'll gather right here. We're going to walk over to the other side of the fences over there. There's a little stand of trees, and I want to show you some microorganisms in the wild to kind of show you what I'm looking for, because I know you guys have all seen them, but I kind of want to identify the specifics. All right, so we go into the forest looking for mycelium. Mycelium is a non-specific term. It does not refer to a species of fungi or a specific anything. It is a catch-all umbrella term saying, I see individual strands, or I see stuff growing that looks like fungus. This is all that mycelium means. So when I say that, I'm just identifying the visual thing we're seeing. And when we go into the forest to do this, we are specifically looking for mycelium that has striations or strands. You guys are missing the most important part of the day. Come on, slackers. No, just kidding. There's nothing about I'm just kidding. Teasing you. When we go into the forest, we're looking for mycelium that have individual strands that you can see with your naked eye, because the human eye can differentiate individual strands that are 4 micrometers or larger, and most disease-causing fungi are 1.5 micrometers or smaller. So if we're identifying individual striations or strands in the material we're looking at, we are very nearly scientifically certain that these are the big beneficials that we want to invite to our farm. Are we afraid about disease? Are we afraid about bringing disease from the forest? Why? Diversity. This is the key. Why? Because most disease microbes function in a vacuum, because nature, wild nature, doesn't have vacuums. Vacuum is the most foreign to nature thing that there is. So disease in nature functions to take out the weak tree in a tree stand so that the other trees have more room for their roots, and so that can be composted, etc. Diseases aren't something that are... I don't necessarily view microbes as good and bad microbes. I don't really ascribe to that, because people are like, oh, E. coli, it's bad. It's like, everybody here's dead without E. coli. E. coli is how we get our food into our bloodstream. If we don't have E. coli, literally, you die. So E. coli is a vital part of nature, just like other, disease microbes, but they play a role in a natural diverse system. They have they're not the main violin chair. They're like somebody with a triangle hitting it they play a role in the symphony, but in a normal, diverse environment, all these big, I describe it like this: if this is our field, we're doing class, and a group of 20 kindergartners came on their field trip, and they're like, "This is our field now," and we're like, That's cute, guys. The adults are talking here. You can be here in a few minutes. Or we say, you know what, throw leaves around. We can take a break. But really, we're going to control that situation because we're bigger and meaner and stronger, right? Sorry, just bear with my analogy for a second. They can't take over by force, I guess is what I'm saying. And the same thing happens in the microbial world where you have diversity. The disease comes in and they go, we'll sit over here and do the one thing you told me to do, over and over again. This is how nature works, and it's beautiful in function. So we're not experiencing as much disease once we get that soil diversity going or the canopy diversity established. Yeah.
Oh, I was just going to say, I think the term in biology is competitive inhibition.
Inhibition. I like it. Good word.
I could be wrong, but
That sounds right to me. I'll look it up though. What? All right. So what we go and do is we look around for where microbes, specifically fungi, are going ballistic. We want large areas of microbial growth. And so this is a small area of fungal growth in the leaf litter here. And what I'm going to do after this is send you out to find some fungi. Because though this is a decent example, we do have individual strands happening in the white stuff. It's also kind of white and fuzzy. It's not a bunch of big old strands. But if you pass that around, you can see I'll pull up some more. But what I want you to go do is just, you don't have to know the exact answer, but I want you to, so you see these strings, these white strings?
Yes.
That white string. What I want you to do is go and look for some of the white strings and bring it back and bring it to me, and I'll tell you, oh no, you found a root, or yes, that's what we're looking for. So you can see it kind of growing on the wood in the individual strands. So yeah, this is stuff we like. Absolutely.
What about this green stuff?
Yeah, probably trichoderma, and has a vital function in nature. You just don't want to choose that to select the
I hear you have the stuff we like.
Yes, we do. It's a nice fungus.
That's nice.
Look at the worm.
So much of it. So looks like cobwebs.
Beautiful.
Can you see the worm? The small worm.
We'll save that as a positive example. We got some more positive examples here.
Oh,
Did you find it?
what did you pick some? Little ones.
Well, that's
Very
supposed
similar,
to do that.
but
Like, like getting orchids or something, or,
easy to tell with a
but you
microscope.
can just take
But
fungus.
these are,
Yeah.
let's go put it next to the. These are roots. These are plant roots.
Oh, yeah.
Okay, those are fungi. These are fungi. These are plant roots.
The fungi is so pretty white.
So no, if the fungi desiccates and dries out, it will get kind of pale in color. But yeah, I don't know. Play with it, feel it. The way the roots feel are different. These are, I think, feeder roots.
How much do you need of it?
Very little. Yeah, so here you go. So a fungi is one diameter throughout its whole body. No matter how long it is, it's the exact same diameter from beginning to end. Roots get bigger and smaller. Okay, so if this was a fungi, each one of those branches would be the same size as the main branch. This is growing from a bigger root into smaller feeder roots. So fungi are uniform diameter all the way through. So yeah, this is a root. Tell people that's a root. He was volunteered by me against his will to be the positive example and now negative example holder. So roots, beneficial fungi. These are what we're looking for, those individual strands. Well, I saw some white fuzzy stuff in the leaves, Chris. What about that? It's good. It's decomposing leaves, but those are not individual strandy things, and they're, maybe think about them as like lower on the food chain than these guys, if you will, or less vital. So they're not bad. There's nothing wrong with them. We just don't want to spend all the time to put a collection box on them and grow them out on a substrate, because that kind of microorganism will come passively very easily once you establish your diverse fungal community. Good find, Nina. Ants farm fungi and work with it, and what they do is they drink the dew that the fungi forms, like it's respiration. Yeah, it's an interesting. They like fungal dew. All right, so if I got my box, this is what I'm looking for to place on, because even though this might not be the exact big strands, what I'm gonna do is, if we all were placing one box, we'd take, dude, I should have got your name. What's your name again?
Connor.
Connor. Can you bring those over? So I will relieve you of your task now. Thank you so much. Oh. So what I do is I'd place my box right in the middle of this big bloom of beautiful fungi, and look at that goodness. So that's probably a mycorrhizal fungi, where it's infesting the roots. So some fungi actually moves into and shares space inside the roots. It becomes alive in the cambium layer of the tree, and it can live all the way up in here. And a lot of disease-causing fungi do the same thing. They get up into the cambium layer and they destroy the tree. They cut off its blood supply, if you will. But beneficials will do that and work symbiotically with the...
and will actually block out the pathogens.
Will protect... Yeah, because they take up the space in the root zone, that good feeder space. So those all in that root system like that are probably some version or some type of what's called mycorrhizal, which means it lives within the kind of within the tissue of the plant's root itself. So what I'd do is I'd place my box, and then I would tuck this all over it and around it. And I'd take one little piece of really beautiful fungi, maybe that string hanging off that root like that. See that big string there? You can see it all the way from the back there. And I'd tuck that in on top of the rice so that it had kind of first access. But there's so many holes in the bottom of my rice box that there's rice falling out onto the ground. It's kind of like the stairway to heaven for the fungi. They just move up into the whole box of rice and bloom in it, okay? So I'd place it here, cover it in these things from the surrounding area. I'd bring over different microbes from the surrounding area. I'd tuck it all on top. I'd kind of pull in my leaves, and I'd leave it here with a tarp that would be up. So a piece of plastic, whatever, I'd prop it up so there's still airflow, because I still want the yeast and bacteria in the trees and everything that's going on in this forest to all infest at the same time, or inoculate my rice at the same time, because I don't want just the fungi, I want the whole community. And then I'd come back, and these temperatures, with it getting this low, it'd probably be 10 days, because once it dips below 40 degrees at night, you can't collect IMO. It'll take forever, it'll never progress. So it needs to be 45 is probably the lowest I'd bother doing a collection, and that will probably take you 10 days for it to get a bloom because it's getting so cold at night that everything stops working. But yeah, once your nights are above 45, you can collect a collection in 7 to 8 days, something like that. If it's 70-degree weather, it'll be about 5 days. If it's much hotter than 70-degree weather, it'll be less than 5 days. But yeah, come have a look at this. This would be what I'd want to find. I'd want to find evidence of abundant life. That's what I'm looking for when I try and find a place to put my collection. How beautiful is this one?
Like the environment's good for it, basically.
Yeah, the environment's good for it.
Happy here.
So the box design prevents rodents. A bear could get it, but rodents won't be able to. Yeah, I do a little like hardware cloth, that 1/4 inch kind of metal. And so I put the box out, paper towel with just an office stapler, staple the paper towel around, then I put the hardware cloth on top of that, and I staple that around. And so the box, there's no holes big enough for a rat on the bottom or anything, and then they can't get through the wire. Yeah. So I have 2 IMO 1 videos on YouTube. Watch the second one, it's better. But you can watch both of them, they have information. How pretty, right?
So if you were to take something like that and say it's like really cold outside and you're going to take it home?
Great question. Winter collections. If it is below 40 degrees, how do I collect IMO 1? Get a bucket of 5-gallon bucket, fill it with half full with this stuff, go home, make your box with your rice, put this in cardboard, kind of create a base just like we'd place a base here, and then tuck some in, put it on top, and now you're in like 70-degree weather, maybe in your garage or in your living room, and that'll take about 7 days. And you can do it that way. The reason we still use the rice is because we don't want any of this organic matter. If you try and put this organic matter with brown sugar to try and go and cryofreeze, it'll turn the brown sugar to alcohol. It'll go off. There's too much bacteria and organic matter, too much activity happening here. What we want is to keep the organic matter out by putting the box contained in this place and get only the rice and the microbes to bloom in there. If you go out and you find a beautiful place like this, all this fungi, and you get a great collection here, this is your well. This is your place to find life. Take care of it. So when you're done, tuck it all in, cover all the leaves back, because that, even though we uncovered it and dug in it a little bit, that will be thriving and abundant if we cover it back up, and it'll be there for years. This is a part of your soils, your forest's abundance, your forest resources. So keep your mycelial mats really hardy cared for, fast-growing plants. So to select an FPJ plant, what you want to do is you want to look around in your environment and find plants that grow. They're green all the time. They always look healthy. The bugs don't seem to bother them much. Nobody cares for them, and they're lush and rich because those plants are being effective at pulling the nutrients out of their soil. So they've found a balance maybe with the fungi to bacterial, but why we want them is because they're nutrient rich and full of oxygen. So they're fast growing. So right now in the spring is the time to look around and see what is shooting up like crazy. You can't kill it with a brick, and it's just hardy. It'll be the plant that you literally don't see. You drive by every day and you don't even see it because it's always there, and you just have it's phased out of your mind. You don't even notice it anymore. So readjust your sight and look around for the weeds that are free and easy to harvest. All right? Blackberry, new growth of blackberries is an incredibly rich in auxins material. But once you make some blackberry FPJ, you will find that you need to find another plant to make for your garden food, because the blackberry FPJ will not leave your pantry. You will use that for human food. It tastes amazing. You're using the new growing tips of all these blackberries. It tastes like blackberry. Yeah, it's really, really good. All right. So stinging nettles, we gather them tight, and we chop them about the size of a $quarter. A quarter. Here, I can take these down. And what we're doing is increasing. You can rip it. Ripping it is even better. You're not creating that knife slice on it. But we're increasing the surface area. That's the point here. So you can whack it. You don't have to do it. You don't want any roots or dirt material on there. So if you're harvesting FPJ material, make sure not to pull up roots. I have a friend that likes the sting of stinging nettles. I think there's something wrong. Yeah, there's a few of you. Strange. Watch out for them. What? There's a little bit. Definitely a little bit. Yeah, it is. All right, so I didn't give instructions to the harvesters of this, so we did get a little bit of roots in here. So I'm just selecting out for the dirt. We don't want any of that in there, or roots. Yeah, looks pretty good now. All right, so we whack all our fermented plant juice material into small pieces. And the next step would be to weigh it. We would tear out our bucket. I did not ask for a scale, so we won't do that today. You can eyeball it, but highly recommend weighing it, because the reason we're weighing it is the same reason we weigh IMO 1 material. We want to account for all of the water in this environment. With brown sugar weight, because though we're fermenting this, we have a very controlled fermentation with FPJ, where we're allowing it to ferment. Some minor alcohols will form that will compromise some of the cell walls and enable us to extract some of the plant's material. But if we use too little sugar, it will just take the sugar and the plant material and make alcohol. Which will then make vinegar. If we use too much, it won't ferment and it'll stay really dry. So the equal weights is important. That is slightly variable. If you had plums or something, green plums for FPJ, they would be rich in water, and you might need a little extra of the sugar because there's more water than plant material or solid material there. Whereas these are a little drier. We might use a little less sugar, but it's generally equal weight with slight variation. You always harvest fermented plant juice materials early in the morning. When do you guys have more energy? In the morning or right before bed? Plants are the same. All day they respirate. They're maintaining their temperature, and they're photosynthesizing. It's like an engine at full speed all day, and then it refuels at night and moves nutrients around. What it does at night is it takes all the nutrients that accumulated in photosynthesis, especially now in the spring, and it pushes all its accumulated nutrients where? Into its new growing tips. So all that fresh growth has all the work and nutrients and biochemical production concentrated in it, and we're getting a really high quality or high value fertilizer. All right, I am going to go based on what I think the weight is, which is a little more than that. And then what we do, yeah, my fingers are going to be tingling all day for sure. I don't care what sugar. Ooh. I'll talk to the sugar giver about what I found. Say, No, it's a vitamin. It just looks suspect. I was like, what is this? And then we do a rough massage. So we're just kind of crimping it a little bit. What?
Like kale.
Yeah. Like a kale salad. Not anymore. I could have in the beginning. I think I'm committed now. Yeah, maybe it'll help me. If I start puffing up, somebody get an EpiPen.
Would you recommend comfrey or burdock?
Absolutely. Both of those are great. Anything that's kind of, both of those are, kind of fast growing, hardy, nutrient rich. And then what we do, I think we're halfway between these two. I'll use this one anyway. I'll use the big one so we can see it. That'll be easier.
I was gonna strain in that one, but.
Oh, you were going to strain in that one? Yeah. Yeah, we'll do this. Yeah, you want to help me, Michael? You want to be the hand? Actually, hold the funnel. We'll make this the funnel. There we go. Maybe we'll just funnel with your hands.
Yeah, that's fine.
So get them like that. Maybe. There we go. So we usually use like a largemouth jar. You can find these at Walmart because it's much easier. But it works in this. You just got to clean up your mess. We'll do one more. You want to get your other one? My hands won't fit in there, so I can't use these.
Yeah, my tiny hands are good.
There's a principle in natural farming of 2/3 airspace. Thank you, Michael. That we use across the board. And I want to explain that because that's an important part. All right, you want to use the paper towels and the rubber bands and put a cover on that while I explain the 2/3 airspace.
Sugar cap?
Let's not do a sugar cap. And maybe wipe off the edges. How many of you guys been camping? Okay. So it's cold outside. You're outside hanging out. You're cleaned up after dinner. Super chilly. You get in your tent. It's warmer, if not initially, pretty quickly. Why? Body heat. Protected from the wind. Respiration, you're breathing in there. In a very short time of laying in there, or a couple people laying in your tent, even though it's a super thin material, you have conditioned that environment for yourself, right? Your body is regulating the temperature in there. If you were a lava monster, it would probably be too hot, right, for you. But you're you, and your body temperature is convenient for your living. Your breathing and respiration is a nice humidity for your life. Microbes are the same. We create a small airspace when we do the IMO 1, where we leave 1/3 to 1/4 airspace in the material. One-third air. because the microbes in the fermentation process actually use that airspace as they start to ferment. There's a unique or a rarefied kind of environment that's created. So specifically, very importantly, in the IMO1 process where you put that box out, you want your rice at 2/3, because what'll happen is they'll use that airspace between the rice and that paper towel and they'll bloom into it. A lot of times if you have a really good collection, you'll come in and you'll see this mound of growth up into the airspace that you left. And so that same concept is happening here. Though there's fresh air coming in, the fermentation is actually fostered by this little bit of airspace being maintained. So generally speaking, we're creating a 1/3 airspace for ideal fermentation. All right. Well, we have a super treat that Michael heard it on the wireless and created a nettle FPJ for us a week ago. So you get to see it finished. So this is, he went out and harvested it early in the morning, got a bunch of nettles in his hands, and is showing this. So
went out at sunrise and with the dew still intact with the plant, and harvested all the growth tips. So the nettle, it was in the field and there was an abundance of it, and the first thing I thought of, because I know nettle's perfect for FPJ, it's very fast growing, it has a lot of nutrients in it, it's just beautiful for this process. And I've worked on a farm where they made gallons of this stuff with nettle from the stanza nettle in California. This just goes for big stands, and you utilize that to bring into your farm. And I woke up early right before sunrise, went over to the farm that had it, and just harvested the growth tips, brought it back home, mixed it with equal parts brown sugar, put it in these jars, and I always like to label and date my ferments. So
always label and date,
because you will forget. You might be really good at remembering, but sometimes if you're having like 10 different kinds of FPJ and 20 different kinds of the other FFJ, you can get mixed up with when it was made, and it's just good to know all that stuff, to know the age of it, and because you need to make it every 3 months you want to make it?
It depends on your farm, what your needs are. I make it every 6 months for my farm.
So basically, now we're about to strain it out, and I put should I explain the sugar cap?
Yeah, so sometimes we do a sugar cap, what that is it kind of prevents initial oxidation on the surface, so it kind of
We'll see in a minute.
No, the things that would be there would have broken down, I would think.
Yeah, it feels pretty nice and smooth now.
So there's no water added. This is just.
There's no water added, and so
all the liquid is the growth hormones and enzymes now that have been basically the
and
juice
the plant's
plant.
liquid, the plant's juice. So we got about that much liquid in it. There'll be a bunch of liquid in here. The longer you can have it sit to strain, the more of the liquid will kind of come out. So typically, once you create it straining like this, you'll leave it here for about 30 minutes. Never. You don't want, so if you just wanted the juice of it, you could put it through a juicer, but that's not the point. We actually get a specific part of the plant out through this process without pressing it, where it extracts a lot of the good stuff without kind of pressing out just the juice. I don't think I explained that very well. Yeah, don't press it. You just
Let
drip
gravity do its work.
We'll do one more like on here. You're all gonna taste it in a second. So all your natural farming fertilizer you can eat. That kind of makes sense if we're making food that we could eat our things that are going on our food. So we there's a whole segment of past students, and they've gotten really into the culinary uses of their fertilizers. It's good fun. I think we better move this one over.
Yeah.
Yeah, maybe move that one to the other side. There you go. Question. You can, but there's better uses. I was giving it to my worms for a while, and I was like, wait a second, this stuff's too good to be given to my worms. It's like all this excess sugar and these like perfect harvested in the morning plants. So I Googled vinegar, and then it had this kind of elaborate process, and I was like, I wonder if you could skip some steps and still get vinegar. And turns out you can. So what we do, once this finished straining, we drip out all the juices. And this, like a lot of plants, produce maybe more juices than you're seeing with this particular nettle. And I don't know why. I think nettle usually produces pretty well. But you take the solids, add it back to a jar, or add it to a bigger container, and then you add equal parts water. And you leave it for 3 months or so, and you'll have a super acidic homemade vinegar that you can use in your vinegar needs for the natural farming process, because we do use some vinegar. So I also make wild wines out of my byproducts from these. So I made a jalapeno champagne. You had some of that one, right? Yeah, and I made a prickly pear champagne.
That too.
That one was good. The prickly pear champagne was probably the best I ever made. It was. I had it all in all these old vodka bottles because that's what I had on hand when I was straining it out. And you'd open it and it'd go boom, and then it would. It was just like a. And then because prickly pear is viscous, the champagne was viscous. It was very strange. Like it was just slightly more solid than water, but it was super tasty. But that was the kind of the point of this conversation about vinegar and making alcohols. Is this is one of my fertilizers, one of my plant foods, an important part of producing my vegetables. But this has a cost that is sugar, right? It's free, and the sugar cost. If I can take that sugar cost now and get a vinegar product, or get a bottleable beer that maybe my friends know I make and they buy for cash or something. Now I'm paying for all the sugar, and now my input costs are free because I make alcohol out of my byproduct, just adding water. You can see how, we're economizing the process of growing our crops.
For the vinegar, when you say equal parts of water, you're talking about by volume?
Yep.
You're covering it, essentially?
Yeah, kind of accounting for what's seeping into the cracks. What's your favorite vinegar you made, Michael?
I made aloe vinegar.
That's good. So we leave this to drip for a bit. I'm going to move this one over, and we're going to make them taste this. You can just put your finger in it.
Thank you.
No, it's good. It's good. It's good.
You're gonna skip it. Oh, yeah. Pregnant, I don't wanna.
That'll be good for you.
I'm sure it would be. Oh.
Yeah, maybe they'll like that. Oh, hell yeah. Is it very sugary?
It is a little bit sugary, yeah. Okay. Listen very carefully. I taught you this, but you can't get it wrong just because I taught it to you. You never, ever, ever use FPJ by itself. The key to natural farming foliar nutrients is balance. it's a balanced nutrient. So all the natural farming nutrients can be taken up foliarly. They're all highly available. So when we buy fertilizer, like conventional fertilizer, and put it on the ground, we're hoping for about a 20% uptake. That's a typical number for conventional fertilizer. With this material, it's full foliar uptake. It can be absorbed through the skin. We do it with fish. We make a fish amino acid. It's a fermented fish. Yeah, it's tasty. Oh, perfect. Oh yeah, you just wait until you taste some I made. So one of my good friends is a fisherman in Hawaii, and he'd catch ahi. He'd go out in the morning and catch ahi, and he'd call me at like 9 o'clock. He's been out, harvested his ahi, taking the fillets off the carcasses, and called me, and I'd go by and pick up all these fresh filleted ahis, carcasses, heads and guts and tails. And I'd be there with my knife cutting off some pieces and eating it while I was making it, but I like raw fish. But then you ferment it with sugar, and you use a little bit of IMO, which is the fungal component, because fungi can process the fats and proteins of the fish so it doesn't putrefy. It smells amazing throughout the whole process. It never goes stinky. And then that material I actually wish I hadn't used on the farm. I wish I had just saved it all for a kitchen ingredient, because it was fresh. Because who's used fish sauce? Yeah, this is a whole lot better than that, because they're not. But yeah, it's just a fermented fish sauce. But because of the microbes' use of this, it's micronizing the particles. If you go and you check out emulsion under a microscope, it's large chunks. Microscopically, it's quite large. But if you ferment it, and all the liquid is created by the breakdown process of microbes, then microscopically, it's a very small particle. So it's highly plant absorbable. So these are all able to be applied on the leaves. So for FPJ, it is used in nearly all of the feeds for plants. It's a pretty essential part of the whole process. And I will tell you how we use it. If you want to know how to use it, what I'm going to explain is in a video called Maintenance Solution. Maintenance Solution, oops, that's an M. So this is FPJ 1 to 500, OHN 1 to 1000, and BRV, brown rice vinegar, or just, if you can't get brown rice vinegar, you can just get any vinegar or make your own. 1 to 500. Where did I put that? Guys, this seems, I'm writing this here, I'm like, this seems complicated. This is actually very simple. The only complicated thing is getting the nuance of making the IMO down. The rest of it is quite simple. It's just like, you saw what we just did. That's the whole process of FPJ. There was nothing left out. You just saw that's a lot of it is quite literally that simple. But the use, slightly more complicated in that there are ratios. I'll explain what a ratio is. But FPJ is balanced by the vinegar, and then OHN has an alcohol base. And so it helps with the vascular, or the movement of nutrients through the plant. A little bit of alcohol but also it has 5 antipathogenic herbs: ginger, garlic, cinnamon, licorice root, and angelica. This is one of the things I sell just because it takes time, and people are like, I want to start doing it. I don't recommend buying anything. I recommend making it. But it does take 2 months. The OHN is like one of the ones. So what a lot of people do is they get together. One person wants to make it, the other person wants some, and they're like, hey, we just double the ingredients, and we all, make it at your house, and you can get together and start it together. It's more fun to farm natural farming with friends. Like having a buddy to do this stuff with is a whole lot more doable. My natural farming buddy when I started was a guy named Poncho. You guys know Neil Young and Crazy Horse? So Crazy Horse is Poncho's band. He's a rocker. He's the Crazy Horse band, and he would do natural farming with me because he wanted me to succeed on a large scale. But it's a lot more fun with friends. So 1 to 500 means if I had 500 gallons, I would use how much? 1 gallon. 1 to 1,000, if I had 500 gallons, I would use how much? Half gallon. Yeah, this is a smaller number. The bigger the second number, the smaller the amount you use. If I have a liter, I would use 2 ml, or 1 ml. Anyways, ratios, the only reason we use ratios is because this is a recipe no matter what volume you choose to use. It's the same recipe for everybody. Yep.
What volume do you make that? Do you have like a big production area?
For my farm?
Yeah.
Yeah, I think I used it was either 32 gallons or 64 gallons a year for 750 acres. It wasn't that much. But I mean, for like a farm this size, the whole thing, you might need a half liter for the year. I mean, it's very little. You're not making this all the time. You're not making a ton of this. You're not buying a amount of brown sugar. What we just made could probably be half as much as they'd need for this whole project for the year. Yeah.
This one has a 33 or 64, whatever it is. I mean, how much did you start with of this raw material? I would get 20 gallons per 200 pounds of combined material. So if I used 100 pounds of sugar and 100 pounds of plant material, I'd get 20 gallons. More or less.
And you'd have someone go out and harvest that?
I would harvest it.
You would?
I did all the natural farming for 750 acres by myself, plus managed. It was a one-person job. I had some friends that helped me chop up FPJ on FPJ Day. Yeah.
Could you repeat what OHN stands for?
Oriental Herbal Nutrient. I have a video on how to make it. I'll write some of my contacts down and some of your research so you can find some of this stuff. But the reason I wrote that up there, that's the base nutrient. If you want to make FPJ and you want to use it and you have nothing else, equal parts vinegar, both at 1 to 500. The vinegar balances the FPJ. Otherwise, it will be unhealthy for your plant. All right, so my website is biomay.solutions. And Instagram at Soil Steward. I also have Biomay Instagram. YouTube is just my name, Chris Trump. And my email is just chris@biomay.solutions.
I'm not going to give you my phone number. Unless you ask. You could ask and I'll give it to you. It's just because I don't answer my phone. You guys are awesome.